Hi everyone, my name is Cohen and I will present now my seminar on that's called Plug and Play PSF Engineering for Color and Depth Encoding under the supervision of Yoav Shechtman. I will just add a pointer. See, there's a pointer. Okay. Yeah. Okay. So we'll start with the introduction to microscope. So what is a microscope? A microscope, we know that we want to look at biological samples or samples in general, but biological samples in high magnification. So a standard microscope, we can look at it as a closed black box. And when placing a sample, we want to form an image and to see it. So a microscope is composed of two lenses. The first one is closer to the sample and it's called the objective lens. The second one is called a tube lens. And we also have a mirror for the purpose of our project. It is simply bend the light, and I will sometimes ignore it for sketching. So what happens, the objective lens collects the light from the sample and through the mirror guides it into the tube lens that then focuses it and forms an image. This is the place where we place the camera and we get back the image. That way we can get nice pictures with high magnification, like this microtubule. I'm sorry, this mitochondria and this microtubule. But there are limitations for the standard microscope. Some of them may sound trivial. So the first one is we can't generate volumetric data. We can see the 3D geometry of the sample. We have to do something for it. It also has a resolution limitation. This means that an image with small features will get blurry and we might lose some of the features and the resolution will be limited. This is a physical limitation. And color, we can't get color without any intervene in the system. So this thesis goal is to design a component that looks like this, like that, that can slides into the conventional microscope in an existing slot. In this project, it's the DAC slot. So it slides into and help us to encode or add the capabilities of these options to the standard microscope. The thesis will be divided into two. The first one we will deal with color, and the second one will deal 3D super resolution. So begin with the first part, color encoded plug and play. First of all, why is color important in microscopy? Here we see an image that acquired by a microscope, and it doesn't have spectral information because it's gray. In this case, in biology, we can add emitters or colored fluorescent molecules, for example, with three different or with multiple different colors that are biologically attaches into different features in the sample. So for this example, if we acquire colored image, you can see that the nucleus is painted in yellow, the mitochondria is painted in red, and the Golgi complex is painted in cyan. So this helps us, or this can help the scientist to differentiate between the different parts of the sample. This can add a lot of information. So how can we encode color in microscopy? First of all, I want to get things straight. We don't use color camera in microscopy because in microscopy there is something that's called photon budget. We don't want to lose light, to lose photons, because because of the high noise, typically low signal and high noise of the system. And color cameras are throwing away a lot of photons. So it's definitely a big no in microscopy. But what can we do? The first and most obvious and easiest things to do is to illuminate the first one, for example, the red color, and grab an image. The second one will be the yellow that will give us the nuclei image. And the third one is the cyan that will give us the Golgi complex. Putting them all together will give us the colored image. But as you can see, this is 3 times longer. And in some cases when we want to scan a large area or to see a dynamic sample, we don't want this to take so much longer because it will add a lot of time to the process. Or in the dynamic sample, for example, the sample will move between 2 frames, so it will be difficult to stitch the photos together. So this will make the clock very sad if we are taking too much time to do so. I will go over 2 more options quickly, just to show the concept and the problems in them. So the first one will be simply to split the light channel. We add a camera, we add some optical hardware, and each camera requires different color of the image. So for every color, we need an additional camera, but that way we can take it simultaneously. Another option is to split the field of view. So some part of the sensor will represent different colors. So this will take some of the field of view and also add some hardware. And adding optical hardware to a setup, it's not easy task to some people or to some scientists that are not that doesn't have experience in it, and this can prevent them in doing stuff. So that's why we want to make it accessible and easy and easier to use to the non-expert in this field user. So we want a component that slides into the microscope and somehow encode spectrum. So for white point emitter, we will get a separation in color somehow. But the question is, what should we put there? So the first option that we can do, and the most obvious one, is using the rectangular diffraction grating. This is a transparent component that has like a rectangular shape, rectangular period in it, and for an incident light, it splits the light into 2 diffraction orders, even more. For our purpose, it will be 2. And each angle is symmetric, and each color gets a different angle. So longer wavelength will get larger angle, and this is some way to encode color with separate color. And so these are called the first and minus first diffraction order. And there is also the 0 diffraction order that, according to geometry, it can be mitigated up to, we can say, non-zero. So let's see what happens when we put it behind the objective, just like our plug-and-play component. So the objective lens will collect the light of the colored image, and the diffraction order will split it into the first and the minus first diffraction order. So the diffraction grating will do it. Sorry. Yeah, the diffraction grating will split it into two parts, and on the image sensor we will get a replicas of the spot. So we will have two replicas of the same spot when the color is splitted, and the distance between the colors can give us the color. This solution may be useful, but it's not the best because first thing, it splits the photons. So we lose half of the photons. Maybe we can do better. There's more problem that we will see next. So another solution, and maybe better in some areas, is the blazed grating. It's periodic, but it has a sawtooth shape, like we can see here. And now, for the same case of collecting light, it concentrates the energy into single order, the first order, that also contains the color information. And again, we get a split of the color, and now up to 100% of the photons remains. Of course, there is some loss of photons, and I'm not talking about that right now. But the problem with this approach is because of the large angle of diffraction, the beam deflects the light into relatively can be large angle in some cases, and it can put our image outside the field of view. That is why usually when using diffraction gratings, it's done outside the microscope with additional hardware. So because it deflects, it might hit the wall of the microscope because again, this is a closed box, or just miss the lens because of the large angles. So we came out with a better solution for this case that we call the Terrace grating, just like the Terrace in biomedical engineering building, inspired by it, not really. We came up with the transparent component that has terrace shape in it, or staircase shape in it. And it turns out that for an incident white light, the color information goes straight and it's not deflect. So the first order goes straight. Specifically, if we design it for the yellow will go exactly straight. The red will go to the left, slightly to the left, the blue slightly to the right, but it will remain close to the axis. And the white light, the zero order, is the one that deflects this time. So let's see. Yeah. So fortunately, we have an analytical solution. It allows us to investigate how it behaves. So intimidating at first, we can separate it into two terms. The first one is the zinc envelope. The second one is the Dirichlet kernel, which is like a periodic comb. Let's see what happens when we draw them all together. So the animation is for changing wavelength. It begins with the green color, 500 nanometer, up to 700 nanometers, which is the red color. And so we can see the envelope, the zinc envelope. Show it, this one, the zinc envelope. And we can see that it doesn't change. It only gets wider, but it doesn't shift with color. And we can see the comb like the peaks. They are the diffraction orders. So we can see here that only one of the diffraction orders is within the envelope, and therefore this is the only one that will survive. This is the first order that we saw earlier. And what else we see is when we increase the wavelength, it goes to the left, slightly from right to the left, and it crosses the zero. So in the yellow part, it's on 0, again on axis. So let's see what happens now when we put it behind the microscope. So for the yellow part, as I said, it goes straight. For the red one, it goes to the left. For the green, it goes to the right. And we get the spectral separation while we don't deflect the beam at all or too much. And we get an image back on the sensor plane that encodes color. The distance between the colors is the spectrum or the color. Fabrications. We used to fabricate it. We used a method that have been developed in our lab by Reut Orange Kedem. It allows us to fabricate it by 3D printing. And if you think about it, 3D printing is the Terrace with geometry is very suitable for the 3D printing because it's straight, flat surfaces, flat staircase, but it contains flat surfaces, which is very suitable for 3D printing. That's one thing. So the workflow or the workflow for fabricating it is we are 3D printing a template. This template has the terrace shape in it. Then we use it to fabricate the transparent element, attach it to the microscope adapter. The adapter is also 3D printed. This is how we got the plug and play component. And we slide it into a microscope. What we see here is the microscope. We took off the objective lens just so we can see it. Then we put the element in it, the thing that blinks it that I marked. We slide it and we will thread the objective so it will be behind it or right after it. Of course, we don't have to thread it away. It was just for the picture. And we use this, we fabricate it as a collaboration with Tel Aviv University, where they fabricated and they designed and fabricated a chip that contains a lot of different microRNA molecules in it. And by applying voltage, they made it stretch. So they have stretched microRNA like that. They added to the sample different fluorescent molecules with different colors, 4 different colors. And for each microRNA type, the sequence that it is attached to is different. So, for example, the first type of microRNA will be yellow, then red, then orange, then green, etc. The second type will have a different sequence. So the sequence of the colors means or equal the identity of the microRNA. And they want to scan a lot of it, large field of view and very fast. This is the application for the research. So let's see what happens when they put it under a microscope, grayscale microscope. The stretched microRNA turns out to be gray, and we lose every color in it. And we can distinguish, we can detect the different barcodes. But what happens when we place the terrace inside? Every color shifts by a little, and now every barcode has a unique shape in it. So the barcode, instead of being a sequence of colors, is now a different shape. And by image processing, they can identify the type of microRNA. And these are the results. So we put it inside, we did a test. First, we did sequential illuminations, means we illuminated with green, then red, then yellow, and took 3 different images, put them all together to see the color image. And we can see here that if the center of the molecule is, if the yellow is not shifted, so the red is above the yellow and the green is below the yellow, and this shape is related to a unique barcode. Okay, this was insanity check. And then we did a snapshot. So we illuminated all at once and got the grayscale image like we see here. So this is, as you can see, this has the same shape as in the sanity check, and we compared it to the simulation to what we expected and what they expected this barcode to be. So this is GADPH. This is another one, pose A. This is how it looks in the simulation. You can see a unique shape for every type of barcode. And lastly, the HIFI1, F1A, sorry, has this unique shape. Again, as a plug and play component, I can tell you that they did it with additional components. They add prisms that they rotate. So this case is very simple, especially for them and for people who can build such system. But this is relatively an easy case. Why do I say that? Because we have a stretched microRNA, so we have like a reference point, and we have a unique shape for every shift root. What happens if we have a single color that is not attached, that doesn't have any reference points to it? Let's see what happens here. We are sliding into the plug and play, and we get a shift according to color. But we don't know by how much we shifted it because we don't have a reference point. If this is the image that we are going to see, we can't understand the color. We can't understand that the right one was red and the left one was green because we don't know by how much it shifted. That's why we came with the idea of the dual order terms. What it is, thanks to the analytical solution that we saw earlier, we know that shrinking the terrace by half. Now the envelope and the Dirichlet kernel looks like this. And as you can see, now two poles, two orders are within the envelope and they survive. And this is the multiplication. Now the left one, the left pole or the left order is the first order that contains color and it shifts by color, but the right one is the zeroth order. So it is colorless. It is the image that we would have seen without the terrace. So we can say that it's white. And this is how it looks like. This is the dual order terrace. And what else we can see from the equation is that the white, the zero order and the first order are symmetrical according to the center. And their distance is the shortest that I think we can achieve. So that's also ideal for putting it behind the objective and not expanding it. And this is what happens when we put it behind the microscope. Now we got, I will do it again. Now we got the dots that with our component, it splits the dots into two, and the distance between the 2 points can tell us the color. And that's how we can encode color with a reference point. We tested it on C. elegans worms, NeuroPAL worms. They are transparent worms that scientists are using to scientists and neuroscience and biologists use it to track and see how different neurons behaves. Each neuron is dyed with different set of molecules, of colored molecules. So eventually each neuron has a different color in it. And different color, they want to detect it. That allows them to track the behavior under different conditions and to track the neurons. So that's why they want to see the image. And sometimes, by the way, the worms may be moving. So that is why they want a single snapshot. They might want it. And this is the sequential illumination that we got. We put them all together and got the color image. This allows us to see what field of view we are seeing. Then we put our dual order terrace to get this image where you can see replicas of each neuron. So each neuron got a replica on the bottom. The bottom part is the zero order. And we focused on three neurons, the green one, AS2, and the VD3, which is blue, supposed to be blue, and the VA3, which is supposed to be red, according to the literature. And we plotted a vertical line, vertical profile of these three ROIs, and put it on the same graph. So we see here two peaks and the distance between the two peaks. Here, this is the first peak, this is the second peak, tells us the color. So the distance of the blue will be the shortest, the green will be slightly longest, and the red will be slightly longest. So that's how we can decode the color of each neuron. Next, I will talk about the next project, taking our knowledge in the plug-and-play component. We want to apply it to something that we are good at, it's PSF engineering and 3D encoding. So first of all, we need to understand super resolution. So the left image is microtubules taken with a conventional microscope. As you can see, the image is blurry because of physical limitation. That's called the diffraction limit, which is a physical characteristic of the optics. So we can't see we can't see small features. But scientists came up with an idea, and they got Nobel Prize for it. That if you can make the sample blink, and you take a single image of the blinking molecules in it, you can detect exactly in a high accuracy the center of each blurry spot, and you can place a pin in it, then repeat second time, third time, and for thousands of frames. And that's how you can reconstruct the super resolved image. This is super resolution. But the problem is this is only 2D information, but the world is in 3D. We want to generate a 3D image, or we want to, volumetric information is very useful to understand the geometry of what we are looking at. To do so, and this is something our lab is very excels at, is PSF engineering. First of all, what is a PSF? Back to the conventional microscope, when looking at a point sample, we get this blurry image. And this blurry image is called the PSF. So when the sample is getting in and out of focus, or when it changes height in its medium, if it's located somewhere in the depth position, the PSF gets blurrier, that's wider and blurry, and disappears very fast. That's why we lose the 3D information very fast. So in PSF engineering, we are putting in the Fourier plane. The Fourier plane is a plane that is located behind the objective or between the two lenses. It's a specific plane. We put there phase mask. What is a phase mask? This is a transparent element that has certain shape in it, just like the Terrace grating we did, but with different shape. So we put it in the Fourier plane, like here, like that. And we get the engineered PSF now. So instead of getting blurrier and losing the information, we get a shape that changes according to depth. So you can see that when it goes from top left to bottom right, and the opposite way when it gets out of focus to the other direction. This allows us to detect the height or the position in space of a certain emitter. How we used it to super resolution 3D. It was developed in our lab again using deep learning method. So knowing the phase pattern of the element, we can generate a synthetic blinking data. This is purely simulated of the PSF, and then we can allow the network to train on thousands and tens of thousands cases where we know the position. So we let the network train to understand what is the position of each molecule in each frame. Then we give an actual experimental blinking data like that with the PSF, with the engineered PSF. You can see most of the molecules here are looking like that. So they are at about the same height, but some of them are like that. So they are different position in space. So we insert the blinking data into the trained network and generate 3D reconstruction, super resolution 3D reconstruction of the mitochondria. This is a work that has been done by Elias and more people in the lab. It's called Deepstone 3D, and it's a work done in 2020. So this is how we use the PSF engineering for 3D super resolution. The problem, the problem here is that the Fourier plane is most of the time is not reachable in the microscope because we can't decide where we want to put the mask, if at all. And more than that, it might be inside the objective lens as a virtual plane, so it mechanically can't reach, unable to be reachable, to be reached. So what we do, we take the camera away and add a relay system that is two lenses that basically does nothing. But now the Fourier plane has a copy or an image of Fourier plane between the two lenses, and we can place the phase mask to there and get our engineered PSF. This is how conventionally we can do it. But this is an image that I took from our lab on how it looks like. So we need to place the two lenses, we need to place the face mask, we need to place a camera, we need to align it. It needs some knowledge and expertise. Otherwise, the image, if we get an image, will get blurrier. And we want to, in our project, in this thesis, we want to redundant this process. We want to make it accessible. We can when we did it, but it comes with a cost. What is this cost? Let's look at this case where we have an emitter at the center and an emitter on the left that is not located in the center of the field of view. So let's see, this is the ray trace. The objective lens also, again, collects the light into the tube lens, into the sensor. And this is how the Fourier plane looks like. It's a beam, so it's a circle. This is how the ray trace of the non-central emitter that is located in the left. This is its ray trace. And what's special in the Fourier plane is that the beam are exactly on top of each other. So here we have two beams on top of each other with a very good overlap between them. So placing the phase mask in the Fourier plane will modulate the light of the two beams exactly the same way, resulting in a PSF, in a good PSF on the center and on the side, on the left. And this actually happens everywhere in the field of view. This is called space invariant system. And this is when we place the mask exactly in the Fourier place. This is not our case. So in our case, there's the objective lens, so we can't reach the Fourier plane. So we put it we put the mask at a distance d from the Fourier plane that we call the mask plane. And in the mask plane, the two beams are no longer overlapped. So you can see this is this beam, the cross section is that one. So placing the mask in this place, in the plug and play, modulates the light perfectly for the center emitter, but poorly or not perfectly for the non-central emitter. So the PSF of the center emitter will look good, but the the non-central one will be aberrated, and this will also happen to every part of the field of view with different aberration. So this PSF will look different from that PSF. And this is a problem because we want to train a small area of the field of view, or we want to train a field of view with these aberrated PSFs. So this is a space variant system. So we modified the imaging model, the non-imaging model. So now, knowing the single mask like before, but adding to it a scalar, the mask distance from the Fourier plane that we call D, will be enough for the model to do the rest. So it will generate PSF that changes according to the exact pixel that it's located in. So again, instead of using a mask, we take mask and D, and we can describe the PSF along the entire field of view. And this is some results. So this is the phase mask that we got analytically, digitally, computationally, with D of 28 millimeters. This is the distance from the Fourier plane, from the exact Fourier plane. And this is the measured PSF that we already saw in this presentation. And this is the simulation. Okay, looks the same, but this is easy because it's the central PSF. But also the top left, this is how the measured top left, and this is the simulation of the measured top left, looks with high similarity. And also for the bottom right with high similarity. So we can describe the PSF across the entire field of view, and we did it. We generated a simulated data, and you can see here that different ROIs has different shape in it. This is the PSF in the bottom left. This is the PSF in the bottom right. You see here like a banana, like something like that, but here you can see like a comet. This is more symmetrical PSF. So we could generate PSF along the field of view and use it for our training data. And we did it. This is the wide field microscope. We took a picture of microtubules area, and this is the blinking data that we can use to generate the super resolution PSF. So this is the wide field. This is the reconstruction without the fork dependence, so it only relies on the center PSF. This is how you would do it without any knowledge to the PSF, and this is how we did the reconstruction with our imaging model. And this is a zoom in to this area where you can see that we detected way more PSF because the network is trained on it on a more accurate PSF in these areas. Lastly, we did it on a Nikon microscope, not Nikon microscope. So this is the 3D printer adapter that we printed. We also we are ongoing doing Olympus, so we can also attach to Olympus adapter. This is the actual adapter, and this is the 3D part that using the magnets of the DIC holder. And in general, we can do it like something that has been done by Nadav from our lab. They threaded it to behind an objective lens. So this will be the most general way to do it. It's not plug and play, but we can do it to every objective, basically. And that's it. Thank you. I hope you enjoyed. Stop share.